Oh my, it's so exciting to be back. And now we're starting a new unit, DNA. Woo, genetics. This is us, guys. This is all about us. Well, and others, but let's find out some little history of DNA. All right, here we have Alfred Hershey, not to be confused with the Hershey, Pennsylvania, the chocolate. Alfred Hershey and Martha Chase. All right, so what they are known for, back in 1952, they worked with what's called the T2 bacteriophage. And here's the T2. This is what the, excuse me, the bacteriophage looks like. It's a virus that actually infects bacteria. So here, this is what a virus looks like. It's got this head and this tail and these leg-like structures for anchoring. And then they take and they insert a probe, kind of, well, you could think of it kind of like a mosquito in the sense of how it just sends that probe down in, and then you can see a little bit of the DNA. So the DNA is up in here in the head, and then it inserts this probe, and the DNA then infects wherever it goes. And in this case, they infected it into a bacteria. And E. coli. So over time, this particular experiment became known as the Hershey-Chase experiment. And so here you can see an electron microscope view of these bacteriophages. So it's not like just one. Here's a host cell, and there's all these bacteriophages that are surrounding that host. Okay, so here's what they did, is they used radioactive sulfur 35 and to label the virus's protein outer capsid in one container. All right, so they mixed radioactive labeled phages with bacteria, and you can see then they took and they agitated it in a blender to separate the phages outside of the bacteria from their cells. And then they centrifuged it, which means they put it in this spinner thing, and it just spins and spins and spins. And so the bacteria, while it's spinning, everything that's any of the solid particles are forming this pellet down here at the bottom of the test tube. And then they measured the radioactivity in the pellet and then in the liquid. Then what they did is they infected, so batch 2 was labeled, so this was with the sulfur 35. Then they did the same experiment with phosphorus 32. So radioactive phosphorus, they did the same thing, except here it was in the protein coat. Here it's actually in the DNA. And they labeled the DNA inside. And then they did the same thing, blended it up, centrifuged it out. And they were able to see that in this case, the radioactivity was in the pellet. And in with the sulfur 35, the radioactivity was in the liquid. And so the experiment showed that the T2 proteins remain outside the host cell during infection, while T2 DNA enters the cell. So what they were able to do is that what they were able to show is that DNA was this transforming agent, and that it carries the information, the blueprint, so to speak, from one generation to the next. So the information is actually carried in the DNA, not in the structure of wherever the DNA is carried. This was a big deal because we just didn't know. We just didn't know. All right, now Erwin Chargaff, he was 1947. So back up 5 years from Hershey and Chase's experiment in 1952. And what Chargaff is known for is developing what's called the Chargaff's rule. And the rule states that for all organisms, adenine is going to equal thymine, and cytosine is going to equal guanine. And so what these are, adenine, thymine, guanine, and cytosine, these are the nitrogenous bases. This should sound familiar to you. These are the nitrogenous bases that make up a piece of DNA. They make up the rungs of the ladder. They're in the center. So on either side, we have the alternating sugar, phosphates, and then in the center, we have the adenine, thymine, guanine, and cytosine. And So if you know the composition of one, you can find the composition of the other three. You can find, so let's say this is adenine is 10%. Well, if I know adenine is 10%, then I know that thymine also has to be 10%. So those would equal. So then I would know that this right here is going to equal, that guanine and cytosine would equal 80%. And so you can figure out, and then of course, if these are 80%, then you just split those in half, and that would be 40 and 40. So you can figure out your composition from just knowing one of those. And we're going to do some math surrounding that. I know there's the word math. Yay! Okay, so let's move on to, so that was Chargaff. He was the one who came up with the nitrogenous bases and knowing their compositions. Now we look at Rosalind. Oh, I like Rosalind. Never met her, but I did like her. She was around in the 1950s, and what Rosalind Franklin's claim to fame is her X-ray crystallography of the DNA molecule. Now, it was with her help, her work, her life's work, that Watson and Crick were able to determine based on what they knew. But they needed, once they saw her picture, it all made sense. So she performed this X-ray crystallography on DNA. And what the picture indicates is this double helix. And this picture would be of you looking down a strand of DNA. So if we're standing up at the top of a piece of DNA looking down, this is what it would look like. Because it's this double helix, it's this ladder that's twisting. And if you looked at a twisting ladder from the top, it would look like an X. All right, now, and of course, these right in here are showing the nitrogenous bases that are in the center. And they're in equal lengths. Watson and Crick were able to look at the sizes here. And they point inward. And you always have one pyrimidine and one purine. The pyrimidines are the cytosine and thymine, and the purines are adenine and guanine. You got to know that, so write them down. The pyrimidine, cytosine and thymine, purine, adenine, and guanine. The purines are larger nitrogenous bases. And so you have two ring structures with them, where the pyrimidines are smaller nitrogenous bases and with one ring structure. Now, if we, so this is where we get the twisting from, because we have these different sizes. If they were all the exact size, then they would just, then it would just be a straight ladder. It wouldn't twist. But you have this twisting because of that. Now, the large areas around the X are the sugar phosphate backbones. So these right here are the sugar phosphate backbones. So those are the sides of the ladder. So alternating sugar, phosphate, sugar, phosphate, sugar, phosphate, with, of course, the nitrogenous base attached to the sugar. And you should write that down, because you're going to need to know that. We're going to practice it, and we're going to draw it and everything else. But you need to know that the nitrogenous base is attached actually to the sugar. All right. So again, here's our piece of DNA. Here's our purines. So adenine and guanine are our purines, and you can see they have two rings to them. And then the pyrimidines are the cytosine and thymine, and one ring structure there. And here's our sugar, our deoxyribonucleic acid for this whole thing. Deoxyribose is the sugar. Here's our alternating sugar, phosphate. So phosphate, sugar, nitrogenous base. And then another phosphate, sugar, nitrogenous base. And they're held together here in the center by hydrogen bonds. All right, Watson and Crick, 1953. They used all of the information that came before them. Chargaff, Rosalind Franklin, all of that information. And they have been credited with constructing the first accurate model of DNA. So they used Chargaff's and Franklin's work to fill in the gaps, because they couldn't figure all the details out. They had some information, but they needed those. It was critical that they had those pieces. So the double helix backbone is composed of, like I said, the sugar phosphate. and and then of course the held together with these hydrogen bonds, and then twisted into this double helix shape. So again, looks like a twisted ladder. Now, don't forget that hydrogen bonds are weak bonds. And what we're going to want to open these up. So when we do replication and when we do transcription and all of that, we open these up. Remember, keep in mind, hydrogen bonds are weak bonds. We want them weak. So we want to open up the DNA during replication and protein synthesis, and we're going to split. This is where it's going to split between the nitrogenous bases. All right, folks, that's all we have for today, and let's move on to the next video because it's getting even better. Bye, guys.